Welcome back friends in another video tutorial from microbiology with Shaknik. So today we will going to talk about a new topic from our protein, that is ubiquitin mediated protein degradation. This video will going to be the part 1 of ubiquitin mediated protein degradation. There will be another video to come. So let's get started with the today's topic, that is the part 1 of ubiquitin mediated protein degradation, where we will going to talk about proteosome. So before going to learn about what is proteosome and what its utility in protein degradation, let's talk about some basic things first. Okay, so the ability of degradation of protein is a very essential function of all eukaryotic cells. Okay, because in cell cycle, if we consider an example of cell cycle thing, then degradation of protein is necessary after completion of one step of the cell cycle. For example, the proteins which are required to complete the anaphase, degradation of them or removal of them is necessary to enter into the next phase of the cell cycle. Not only that, there are various type of protein whose degradation at the right time is very important to survive, right? So that is why protein degradation is important, right? Not only that, if some kind of some kind of mutation observed in the DNA level, then from DNA RNA will be produced. Although between the DNA and RNA formation there should be DNA repair systems also, but if somehow DNA repair system is unable to detect and repair that particular damage will be carried at the RNA level, and then it will be transferred to protein level. That ultimately leads to generation of a protein which may be non-functional or may be capable of doing some kind of function which may be harmful for our cell, right? So what we can do now, we can do degradation of that particular protein, right? And for this type of things, protein degradation is very important, right? Not only that, if you have followed my major histocompatibility complex playlist, then I hope you know that in case of MHC class 1 molecule and MHC class 2 molecule, both the cases, in both those things, the processing of antigen is nothing but degradation of the protein, right? We have also talked about proteosome in case of the endogenous pathway associated with MHC class 1 molecule, right? So you can understand how much important protein degradation is in various type of aspects of biological sciences, right? So it is very important for eukaryotic cell to able to degrade proteins, right? Never necessary, right? So the two main proteolytic system which are present in any eukaryotic cell for the degradation of proteins are lysosomes, number 1, and proteosomes, that is number 2. So first, lysosomes. We have talked about lysosome in our medical microbiology videos various times, right? So lysosomes are membrane bound organelles that contain acid hydrolases. Along with that, they contain some proteolytic enzyme. Both are important for the degradation of protein. Although today we will going to focus on our proteosome, okay, not lysosome. So if you focus on this picture, then you may find lysosomes. This is this one. This thing is the lysosome. Okay, now let's move on to the proteosome. So first of all, before going to talk about this proteosome with the help of these notes, I want to talk this entire concept with the help of some picture so that you can understand it very well. So I am going to use this picture, and I have taken this from CSIRNet Pathfinder Life Science book, and you can also refer this book to understand this. So this proteosome kind of thing, are present in both eukaryotes as well as prokaryotes. Okay, so in case of eukaryotes, these proteosomes are present in both cytosols as well as nucleus. Okay, And in case of eukaryotes, these proteosomes are divided into two groups. One is 20S core particle, another one is 19S regulatory protein. Okay, 20S core particle are single and 19S regulatory protein may be 1 or 2 in number. Okay, so now this 20S core particle, if you focus on this picture, this 20S core particle is that this thing. Okay, this 20S core particle consists of total 28 subunit. Okay, now this 28 subunit are arranged in 4 rings. Okay, and as you can see, each ring contains 7 subunits. Okay, and these rings are stacked upon one another. That's how the entire 20S core particle is constructed. Okay, and interestingly, this 28 subunit all are from one single superfamily, which are divided into two families, alpha and beta. Okay, so that is the idea of our 20S core particle present in the proteosome in eukaryotes. Okay, and another important part of our proteosome is 19S regulatory protein. Okay, this 19S regulatory protein again consists of 19 proteins. Okay, and they are divided into two sub-assemblies. Okay, one assembly is 9 subunit base, and remember this 9 subunit base are actually interacting with the alpha ring of the 20S core particle. This is the 9 subunit base. Okay, and the remaining 10 subunit will form the structure called lead. Okay, so 19S regulatory particle consists of two things, lead and base. This base consists of 9 subunit which are attached with the alpha ring of the 20S core particle, and lead is consist of 10 subunit. Okay, so now if this 19S regulatory particle binds with the 20S core particle from the both in, then we will get 26S proteosome. Okay, and if it bind with the 20S core particle only from one in, then we will get 30S proteosome. Okay, so that is the idea of our eukaryotic proteosome. Okay, now let's come to this slide. As you can see, two 19S subunit attached with one 20S core particle that ultimately gives us 26S proteosome, right? Now there are various functions that 19S subunit performs. First of all, 19S subunit binds with the polyubiquitin chain, and remember it very carefully that this polyubiquitin is very important for the degradation of protein. If this polyubiquitin was not present, then degradation of protein will become very difficult. Why? Because polyubiquitin here act as a level by which cell can understand which protein it have to degrade and which not. Okay, that is the importance of polyubiquitin. We will going to talk about this polyubiquitin and also how this ubiquitination is going on in our part 2 video of ubiquitin mediated degradation. So now this polyubiquitin chain will bind with this 19S regulatory protein, okay, of proteosome, and by binding this 19S regulatory protein ensures that yes, I am degrading a polyubiquitinated protein. Okay, and now not only it is the only function of 19S subunit, but also in this 19S subunit of 26S proteosome there is a presence of isopeptidase. Okay, and this isopeptidase will going to cleave this ubiquitin chain so that they can be used, or I should say so that they can be reused in for some other proteins. Okay, and the third and the last function of 19S subunit is to unfold the protein and to direct them inside the 20S core, because remember the ultimate degradation will be performed in the 20s core particle. Okay, and this type of protein degradation is obviously ATP dependent process. Okay, that is why cell only degrades those proteins which are somehow damaged or which are somehow becomes cancerous. Okay, so that is the idea, right? And how cell recognize those type of altered protein? They recognize this with the help of this polyubiquitin chain, right? And that is why polyubiquitin chain is very very important, right? And how proteosome recognizes polyubiquitinated protein? With the help of 19s regulatory protein of 26s proteosome. Okay, so that is how proteosome can do its job with the help of ATP. Because remember, the degradation of protein in proteosome is ATP dependent process. Okay, so that was our first video of ubiquitin mediated degradation of protein. So I hope this video will be helpful for you, and I am just providing the slides so that you can use those slides to make notes for your own purposes. So let's take a screenshot if you want. So we have also discussed about the 90s regulatory protein functions. So here I have written that it catalyzes ATP dependent degradation of polyubiquitinated proteins. That what I have just explained you. So that is the idea. So that is the end of that video. So I hope this video will be helpful for you, and in the next video we will going to learn how ubiquitination of that protein is occurring inside the cell. Okay, so till then, bye bye.